Hi, everyone. Good morning or evening, depending on where you're located. Thanks for joining us for our webinar on cilia. We will have two talks today. Our first speaker will be Dr. Helen Wilsey, and I'm going to turn it right over to her. So thank you, Dr. Wilsey.
Great. Hi, I'm Helen Wilsey. It's a pleasure to join you today. I am new to your community, so it's a pleasure to introduce myself and tell you what my lab has just started working on in the context of the gene that you are all interested in. So I'm excited to share where we are and where we're heading. So a little bit about me. I'm an assistant professor at UCSF. I have a laboratory there now for about 2 years. And I'm also an investigator with the Chan Zuckerberg Biohub in San Francisco. So my lab uses kind of an untraditional model, frogs, because we want to study so many genes that have been recently identified as associated with rare disease. And in particular, my lab focuses on genes that have been associated with autism. And so this graph here lists some of those genes that we study, but really we study about 250 genes all at once. Your gene is on our list, is number 77, which is why I have started to think about it. But you can imagine that because I'm not just studying one gene, we're studying 200 genes, we need a creative model system to be able to study them all at once. And so that's where the frogs come in. And in particular, the species that I use is a diploid species of frogs, meaning they just have one copy of their DNA from mom, one from dad. So it's a lot like humans. They're really high throughput. They lay over 4,000 embryos in a day. So we have a lot of animals that we're able to manipulate and study gene function in even one day. They develop extremely rapidly. So within a week, you go from a one-cell embryo to a tadpole that's swimming, eating, behaving that we can study. Unlike rodent models and human cellular models, they're very cost-effective. So my animal care costs are orders of magnitude smaller than other model systems. So again, we can study many genes and not have to invest so much in any one particular gene, though of course we can. And genetic manipulation is really great in frogs too. So we have this cute trick where we can actually just make half of the tadpole carry a mutation of interest, like the right half, and then compare it to the other half of the animal and really start to understand what the function of that gene is in any tissue of interest. And the other really great thing about frogs is that we're able to, in a high throughput manner, again, do drug screening. So we're able to actually just put the drugs in the water with the developing embryos and then understand what that drug did during development or even later, and try to identify small molecules that might be able to mitigate some of the effects of these gene variants. Okay, so that's the general gist of our lab. We study many genes. Your gene is one of interest to us, and we do it in frogs. And so to cut to the chase of why I'm interested in cilia, I didn't mean to study cilia, but in studying some of the top 10 of these genes associated with autism, two of them I'm just showing here, which are CATNAL2 and DYRK1A, we started to notice that they were localizing to cilia and functioning on cilia. And so this one here, CATNAL2, is one of the first autism genes that was discovered. And so you'll see these hair-like projections off of the surface of one cell here, and this is in the frog skin, our favorite model system. And you'll see this green here is this gene, this protein product of the gene CATNAL2, and it's in these like small little dots all along the length of cilia, those hair-like projections, but then also down here at the base. And this base of cilia will be important to think about in the next talk you hear. And then we did the next gene on the list, which was DYRK1A. And again, we saw the same thing. There it is out on the axoneme or the hair-like projections of the cell. And then also again on the base of the cilium. So that really sparked my lab's interest in cilia and ciliary biology. We've taken it quite far now with over 30 of these autism genes we've studied, and a lot of them are on cilia. So I want to take a step back now and tell you a little bit about cilia what they are, their history, why you've maybe never even heard of them, and give a little bit more intro about why we think they might be relevant. So cilia are hair-like structures present on almost all cells of the body. They were originally, they were the first cellular structure called an organelle to ever be described in 1675 by Anthony von Leeuwenhoek, who made the first microscope. And he actually described them on unicellular algae. So you can see down here, they have flagella, right, that move and beat and move the algae. And so flagella and cilia are interchangeable terms. And so the word cilium actually comes from the Latin word for eyelash. So you can think about that, right? They're these kind of hair-like projections that come off the surface of cells. And of course, their first described function was a motile function. So they actually beat and they're moving algae, and they're present all the way back to unicellular organisms like algae. Of course, they've diversified quite extensively over evolution, and they're now ubiquitous among plant species and animals. And one of the examples of the diversification that I like is in this picture here of human sperm, which of course their tail is a modified cilium and it beats and it's motile. But within the human female fallopian tube, and you'll see these hair-like projections here on cells in the fallopian tube that actually move the egg down the fallopian tube. And so you can see both in male and female reproduction, these motile cilia are essential. And so again, these are ancient structures. They actually were thought to be vestigial, which means they have no function anymore. We just carried them along from when we were algae. But of course, as you could see in that the example from the sperm and the fallopian tube, they're very much functional. But those are really motile cilia. So not all cells have motile cilia. Certain cells have motile cilia. And so this is one cell here, and it puts up about 150 to 200 cilia that are actively beating. And so this is very much like the cells in the respiratory tract that actually have very crucial functions in moving mucus and controlling mucus production. Like I showed you, that a sperm has one of these that beats. These motile cilia are also very important in heart development. So in the very early embryo, there are motile cilia that beat and actually determine the left versus right axis of the entire embryo. And if there are issues there in the motile cilia, that will lead to heart defects later on in life. They're also really important for brain development. And in particular, this cell type is present in the ventricles of the brain and actually functions to move cerebral spinal fluid throughout the brain. It seems to be very important there for ventricle biology and yeah. Then almost all other cells in the body have what's called a primary cilia. So this is much more like a little antenna that comes off a cell. And this cell in particular is a human neural progenitor cell, so like a human stem cell in the brain. And this little pink antenna here is a primary cilium. It is small but mighty. It is important for a lot of different functions, both in development of many different organ systems, and then also in neuronal function in general. And so the next talk you'll see will talk a lot about primary cilia and really exciting work that has been done there in the context of your gene. But just briefly, this little organelle in kidney development was actually where it was first realized that these structures were important. So like I said, up until the 1990s, it was thought that cilia, these primary cilia, really didn't have a function. They were just kind of a carryover from when we were once algae. But of course, if we've maintained these structures over evolution, it's likely they have a function. And so what happened was in the 1990s, a few genes that were associated with a devastating kidney disorder, polycystic kidney disease, and they were localized. These proteins go specifically to the cilium. And what they then realized was that those proteins in the kidney are actually sensing fluid flow through the kidney tubules to regulate kidney development. And if if the cilium is not there to sense the actual physical fluid flow through the kidney tubules, the kidney keeps growing and grows these awful cysts all throughout it. And so that was really the first understanding that these cilia have very important roles both during development and for regular maintenance and function of human organs. Since then, it's also been well established that this little antenna, which sends and receives signals, that's why I keep calling it an antenna, it's big into signaling, it's really important for brain development. And so major developmental pathways rely on this structure to be able to signal, and in particular, one that's really important for the growth of the brain, which we call neurogenesis. They're also really important for migration of neurons. And so, for example, there's a group of neurons early in development that migrate from the head region of the animal down into the gut and actually form the neurons of the gut. And if there are cilia defects that go on there, that really affects the migration of those neurons into the gut. It's also really important for neuron activity. It's thought that the cilium regulates the balance between a neuron firing and a neuron stopping firing. So it's really important for neuron activity. And I'll show you in the next slide, there's certainly a lot of epilepsies that are known to be caused by cilia defects. And then again, like I said, key developmental signaling pathways, so especially things that control limb formation, digit formation, dentition, which I think is relevant to this crowd, and many functions. So they are small and mighty. They come in two flavors. The next talk is mostly going to talk about these primary cilia. Today, I'm going to talk about these motile cilia very briefly, because again, we're just getting started. But just to highlight this idea that cilia defects, if they're present in an individual, they can cause a multi-organ spectrum of clinical presentations. And in many cases, it depends on whether motile cilia, non-motile cilia, or both types of cilia have been affected by the genetic variation. And so, for example, with motile cilia, what that ends up presenting sometimes, depending on if that gene is required in that tissue of interest, but for example, it can cause hydrocephalus. And so again, that's thought to really be about those multiciliated motile cilia that line the ventricle of the brain. And so that can cause ventromegaly and hydrocephalus. There's especially a lot of chronic respiratory problems in motile cilia are disrupted. And so that shows up like chronic respiratory infections, very serious issues with mucus clearance, and things of that nature. Infertility, you saw the sperm and fallopian tube examples. So if those cell types don't have motile cilia, that can cause fertility issues. Both types of cilia are implicated in congenital heart disease. Like I said, those motile ones early in the embryo, and organ laterality, because of course, again, the cilia are really important for left-right development of the early embryo. Non-motile cilia, which will be the focus of the next talk, and those cause a lot of different things. So certainly, like I said, it's important for brain size development. So you can see microcephaly, corpus callosum issues, molar tooth sign on an MRI, epilepsy, ataxia, intellectual disability, a lot of facial anomalies. The actual development of the facial bones come from a cell type called neural crest, and they are migratory. So like I told you before, the cilium is really important for migration of different neurons to different tissues. And so the face bones and facial structures actually come from a neural-derived cell type that then migrates and fuses. And so cilia defects can often disrupt that process of formation of the facial structures and lead to facial anomalies. I told you about the kidney, the liver, hypogonadism, so undescended testicles is often reported among individuals with cilia issues, skeletal anomalies, scoliosis, extra digits, long digits, obesity. So actually, cilia really regulate a lot of feeding behaviors, and especially satiety. So knowing when to stop eating. So there's certainly evidence for that. And sensory issues, so hearing loss, blindness, and smelling issues as well. So again, cilia, again, they're in all cells. So depending on if the gene plays an essential role in cilia in that particular organ, this whole spectrum of issues can come. And I'm taking the time to walk you through each one of these because, what I would like to talk to your community about more is the spectrum of clinical presentations that you see in your kids, because oftentimes that can give me clues to what organ systems to be focusing my attention in the lab in. And certainly that's been the case for other genes we've studied. A parent will come and say, our kid's mucus is, and that gives me a lot of hints about where to start looking in terms of motile cilia versus non-motile cilia. So again, and people forget this a lot of times, even scientists, and that all neurons have a cilium, and they're actually still a bit mysterious among the scientific community about all the things that they might do. But they certainly have a lot of roles in signaling and controlling neuron firing and integrating sensory signals. There's a lot of different signaling systems that rely on cilium. So these various different pathways, somatostatin, feeding behavior, dopamine even localizes, a receptor localizes to cilia specifically. So I think we're very much scratching the surface about what these primary cilia do, and particularly in the brain. Okay, but let's get back to my favorite model system, the frog. And so one of the reasons that I haven't studied these as much as I have the motile cilia is because motile cilia are really beautiful and easy to study in Xenopus. So if you were trying to do this work in human cells, to get these kinds of cells takes about 40 days in culture and very expensive, whereas I have 4,000 of these animals 2 days after fertilization, and they're perfectly patterned and in vivo model. So this is a 2-day-old tadpole. That's its eye and its head over here, its brain and spinal cord is here. That's its tail. If we zoom into its skin, now you see my zoom background, but also this beautifully patterned skin population of cells where there's this regularly repeating cell here, which are the multiciliated cells. And then there's these other supporting cell types nearby. And so this is very much like what an airway epithelium or the tissue that's in the airway looks like in humans even. And you have these regularly spaced motile cilia, and these cilia are hanging out and actually beating really fast all the time, just like in the airway, moving mucus. There's actually a mucus layer over this entire tadpole, just like in the airway, protecting the skin of this tadpole from infection, just like we do in our airways. So this is our go-to model for studying cilia, both because it's cheap, it's fast, and these cilia are extraordinarily large. So what you may appreciate later on is that cilia are very small in mammalian cells. They're small but mighty, but they're very small. And so that makes the imaging orders of magnitude more difficult to say, is my protein at the cilium? What happens when I lose my protein? What happens to the cilia? It's just more difficult because they're small, whereas these cilia are gigantic in comparison. And so when we start to think about a gene and what its role in motile cilia is, the first thing we do is add a fluorescent tag to it called GFP, and just say, if we look at these cilia and we add in the human protein with a tag, is it even at cilia? And so we've done that for CSNK2 alpha 1. And so we put this tag, this green fluorescent protein, onto the human version of CSNK2 alpha 1 and put it into the frog skin, and there's that cilia. And lo and behold, yes, it does localize to cilia in this system. And so, like I showed you for those autism genes I showed you at the beginning, we see a very similar localization pattern, which is that it's in these dots all along the hair-like projections, which are called axonemes, like this. And then it's also in these foci at the base of the cilium called basal bodies, which you'll see in the next talk, has also been described in primary cilia very beautifully. So it's there. It goes to the right place, and given what's known about its function in primary cilia, I would put some money that it probably has a function in motile cilia. There's a lot of work now that needs to be done. So, for example, we're adding in the human protein here. It may not normally be there. And so that's one of the things to first check is normally, is it on this cell type? Does it go to these structures? And then, of course, if it does, then you can remove it and then say, are there any issues in the formation of these structures or their beating of these structures and things like that? And so one of the things that we could look at too is if you all have patient medical record data, for example, in Citizen or in Simon Searchlight, we could start to look and see if there's evidence for respiratory illness in this population to try and understand if there might be something to the idea that these respiratory cilia might be disrupted. So that's the sum total of what we've done on your gene so far in frogs. So we're excited to take this on more and interested in collaborating with any scientists that might be interested in thinking about this in the context of motile cilia. But I'm going to switch gears a little bit to some clinical biomarker work we're doing in the context of autism and these large effect genes that have been associated with them. But it's the same kind of idea. So like I said, there are these cilia in the respiratory system, and if there's issues of those cilia, can you pick that up clinically? Because there are actually a lot of genetic cilia disorders where the kids have very severe respiratory problems. And so there's actually a clinical biomarker for ciliary function. And so that's called nasal nitric oxide. And so basically in Europe, this is considered a diagnostic test for cilia problems in the context, particularly of primary ciliary dyskinesia, the cilia defect that primarily affects the respiratory system. And so the basis behind this is this idea. I'll show you this one real quick just so you can see the idea. The idea is so that when these cilia in the respiratory tract are normal, they're beating, like I showed you before, and that actually produces nitric oxide gas. Not really understood why that happens. It's something we are working on in the lab in collaboration with Jeremy Reiter's lab. But suffice to say, if there's a problem with the cilia, that nitric oxide does not get produced nearly as high. And so then that's been used now for, I'm going to go back. That's been used now clinically, at least diagnostically even in Europe. But in the US, you're allowed to look at it in the context of research. And so there's a portable machine. It's about the size of an iPad. And you hook up a tubing to it, and then you superficially place it inside the nose of the person that you want to measure for 30 seconds. So it's a non-invasive 30-second test that will measure levels of nitric oxide coming out of the nose. And at least in these severe cilia disorders, there's very low levels of nitric oxide that will be produced from these patients compared to unaffected individuals. So we started doing this for some of our genes that we had localized to motile cilia and had established that they're functional at motile cilia. And so one of those genes, maybe surprisingly, was SYNGAP1. So despite its name, which is so synaptic, it has synapse in the name. Despite that, we've characterized a role for this protein also on cilia and on motile cilia even, particularly at the base of cilia. And so we got an IRB at UCSF to test patients with SYNGAP1 variants. James Schmidt a fantastic technician in the lab, took this upon himself to figure out whether there was anything to this and whether we could actually say individuals with variants in a cilia-related gene have lower nitric oxide. And we went to the patient summit, and he tested 22 pairs of individuals with a pathogenic SYNGAP1 variant and also an unaffected family member matched to that individual. And what he saw was a significant reduction in nasal nitric oxide in the patient with the variant compared to their unaffected family member. So to us, this is strong evidence that this gene is required for ciliary function, given that nasal nitric oxide is such a specific measurement related to ciliary function. It's not related to general respiratory function. Kids with cystic fibrosis have pretty normal nasal nitric oxide levels. So we think that this is compelling human data to show that cilia are relevant in this population of individuals. We have started in CSNK2 alpha 1. We have had 4 patients so far. One really showed a dramatic decrease in nasal nitric oxide compared to the family member, but the other 3 not so much. And so that's why I'd be very interested to know if this population has a lot of respiratory or mucus issues, or if maybe they don't, and that this will be an example where maybe primary cilia are affected, but motile cilia or even just the cilia that are in the respiratory tract aren't affected. So we're definitely interested in recruiting more individuals with these variants along with either a sibling or a parent. Everybody has to be over 2 years of age to do this. So if there are any families in the Bay Area that could come out to UCSF, we would love to have you out. James is also a little bit on tour. And so he's going to Kansas City at some point this year. He's going to LA in December. So if you have a hub of families in any particular city, we can put James on the road, update our IRB, and test individuals there to see if this metric might be informative for your cohort as well. But again, it's a 30-second non-invasive test. The machine tells you if it's screwed up, if somebody, I don't know, if the thing came out of the nose, it'll tell you. So it's very nice in that way, especially with kids that maybe have some behavioral differences. They tolerate it pretty well. James usually brings an iPad with something fun on it and some toys. And so far, it's been really wonderful that both the affected individual and the family members have had no problem completing the test. And so it's kind of the dream test of a non-invasive, quick thing. So we'll see if it holds anything. Who knows? So with that, I'd like to thank my lab at UCSF. Again, we're just getting started on this gene, but love to introduce myself. We're definitely looking for collaborators. The next speaker will tell you a lot more about this gene in cilia and in primary cilia. So we're excited to learn a lot from him. And if anybody is interested in that nasal nitric oxide study, the person to email is James. His email address is right here. If you're in the Bay Area, he can get you into the lab anytime. And then if the foundation wants to come up with a location where there might be a hub of families, we can always send James out and look at it that way. So thank you for your attention. It's really a delight to start to get to know the community and start to see if there's anything we can do in the lab that could be of help. So thanks for your attention. Please reach out if anyone wants to get to know me or chat, or you're in the Bay Area, you want to come by the lab. Please get in touch. My email's here and our website's here. So thanks so much.
Thank you, Dr. Woolsey. That's awesome. And yeah, I will say we were lucky enough to have James in Denver earlier this year to collect those few samples. And as many of you could see from that graph, right, there was only one family that showed a difference, but that doesn't mean much just yet. We really need a lot more families to participate because there could be, you know, a specific genetic change in that family that's different from the others. So if you are even just visiting the Bay Area for vacation, James has been really amenable to making people's schedules work. And then maybe I'll email James, Dr. Woolsey, this week just to see where he's already going to be, and maybe we can tag along
Yes,
on
perfect.
that.
Yes.
Since we do have Dr. Wilsey live, if you have a question now, please feel free. There is a Q&A box, or you can put it in the chat, raise your hand, whatever works best for you. If not, we will go to our next speaker, which is pre-recorded. We just wanted to wait until August because Dr. Wilsey was doing some great work like you saw here. So Dr. Lucio recorded this for us back in February, and he continues his work. So I'm not seeing any direct questions here, Dr. Wilsey, but if I get some after, which I'm sure we will, I will send you an email, but we greatly appreciate your time this Sunday morning.
Thanks for having me. It's a pleasure.
Yeah, and then what I'm going to do now, everyone, is I'm going to share my screen for our recording of Dr. Lucio. And one moment.
Just making sure our audio is going to work. Okay. And if someone can just put in the chat if you can hear this when I press play, just to make sure it works.
Hello, everyone. Thank you for the CSN Kickoff One Foundation.
Okay. Can you guys hear that? Yes, perfect. Thank you, Dr. Wilsey.
Jennifer as well. At the University of South Dakota. So my lab is primarily present at Support Research, which is a new institute. It is about, I would say, it is 10 years old so far. So I joined this institute in 2022. So my lab is pretty new. And so I wanted to just start with the team that I have and I assembled over the past year. So we have few members that are working on different projects. And so one of them is basically OCNDS. And so we are a cilia lab. So we study mostly primary cilia, which is a sensory organ, and I'm gonna allude to that later on. So we have a number of collaborators at Support Research, as well as at teaching with Sampa, who gave us the OCNDS patient cells. We're also collaborating tightly with the foundation. And I think the connection that we have had with the foundation was amazing. We're amazing, actually, and we're trying to expand that as much as possible. And our goal is to try to understand this disease and the underlying molecular disruption there from the lens of the primary cilia aspect. So this is my team. I'm gonna just put the laser on. So this is my team. This is Support Research. This is the north side. And this is a QR code for the lab website if you're interested in looking at different aspects that overlap. So I want to thank in advance the basically the institutions that are funding our work, which are Support Research, as well as NIH funding through the R35 that we got last year. So I want to just start with a brief introduction about the primary cilia and cilia overall. So we know that cilia is ubiquitous, is an ubiquitous organelle, and it comes with different flavors. It has critical functions, and it's widely present in our body. So in the eye, for example, we have these nice specialized cilia. We call them photoreceptors. They are actually cilia. In the trachea, we have another type of cilia, which we call motile cilia. So in our trachea, you can clearly have these cilia that will allow the movement of mucus as well as bacteria along the trachea itself. And so during development, you also have this sensory organelle on the top of the cell. So you can imagine it's kind of a small stick on top of the cell itself. So the cell, this is just like an electron microscopy image, and these are fake colors, of course. But you can see clearly that there is like a small antenna on top of the cell. Of course, you don't see what's inside the cell. You're just looking at the surface here, and the antenna is just sticking out of that. So it's a very cool organelle if you think about it clearly. It's like a great way of detecting outside or the environment, the microenvironment on top of the cell itself. So, and of course, we're talking about OCNDS, which has a broad range of neurodevelopmental phenotypes. Well, in our brain, we have these cilia, and they are also present in our neurons. And this is just showing some cortical neurons of the mouse model. And you can see clearly in red these little antennae coming out of the cilium, of the neuron here. So the neuron is stained with green, and then in red you can see the cilium itself. Of course, I will be happy to respond to any questions further on. I understand from my communication with Gabrielle, it seems that we will have the chance to respond to some of your questions, either online or virtually. So yes, feel free to ask questions. So why this organelle is so important? It is just related to the fact that when it's disrupted, well, it's going to lead to a wide range of human disorders. And so usually there are so many of these, what we call ciliopathies, so diseases that are related to cilia. And it's affecting a number of organs. It's affecting the eye. Wherever you have a cilium or primary cilium, well, that tissue or that organ will be also affected. So we are talking about the sensory organs that we have, the eyes, the nose, the ears. And so these will have also cilia. Also, the skeleton will be disrupted through abnormal development as well there. The kidneys, the livers, and so most importantly, we're interested in the brain. So what are the different phenotypes that we see in the brain in these ciliopathies? Well, it's coordination issues, motor coordination issues, epilepsy, mental disability, brain malformation, autism-like spectrum disorder. But however, we don't know exactly how the primary cilium is disrupted in these different ciliopathies and how it's affecting the brain function. So that's exactly one of the aspects that my lab is investigating. So just like to give you a prevalence number about how widespread these ciliopathies. Well, 1 in 2,000 people is estimated to be affected in these ciliopathies. It's still a rare disease, but it's quite it's often seen in the human population. So the second aspect I would like to highlight is the fact that primary cilia are essential for embryonic development. So if you remove the organelle early during embryogenesis, this will lead actually to lethality that happens usually at mid-gestation. So you can see here clearly that if you have a primary cilia, the embryo is, this is mouse embryos. These are, and you can see clearly that these embryos are normal. And if you remove the primary cilium by knocking out one of the cilia assembly proteins called TTPK2, which is a critical protein for cilia function, if you remove primary cilia, these embryo will die at mid-gestation and will no longer develop correctly. So why this is the case here, and so through the literature and the discoveries that happened the few past dozen of years, we found out that primary cilia are important in regulating signaling pathways. And one of these that are important for embryonic development, one of them is what we call the sonic hedgehog pathway. And so I'm showing that some of the regulators or proteins of this sonic hedgehog pathway is enriched and regulated by the primary cilium. So you can see here, this is a cilium in magenta, the centrosome from which emanates the primary cilium. And so you can see clearly that GLI2, which is transcription factor, important for inducing the expression of specific genes, is actually enriched at the tip of the cilium. Without this enrichment, the transcription factor is not well processed, and it's going to induce disruption of the sonic hedgehog pathway and may lead to lethality. Also, there is this receptor called smoothen or GPCR. It's also rich throughout the primary cilium, and this localization is also critical in regulating the sonic hedgehog pathway and the correct embryogenesis. So besides all these nice aspects and the importance of the primary cilium itself, well, this is a very dynamic organelle. So, and the cues that trigger these dynamics are still poorly understood, and that's one of the aspects we're studying in the lab. So you can see clearly, for example, here, this is a nucleus, so that's the DNA of the cell. You can see the primary cilium here in magenta, or like in cyan, sorry. And so if we go, the cell will actually divide. So one mother cell will divide into daughter cells. And so you can see clearly that the cilium will disassemble and reassemble again later on. So let's follow this cell. So you can see clearly the cell becoming yellow, and then it's going to divide at some point. The cilium is no longer there, and then it's going to reform again. So you can see clearly there are dynamics happening. I can go further with another example where actually the cilium has a base and a tip. And I showed that through the previous images, but you can see in these videos that actually the tip has some dynamics happening there. So you can see that there are like excisions that will happen, and that excised tip will be just released into the media. We call that ectosomes or breaks. This is another example of that, where you can see clearly that the tip is excised as you go. And this is an active phenomena that we see and we're studying, trying to understand, and it could be disrupted in ciliopathies like the BBSOM or like the BBS syndrome that are also ciliopathies. So another way of also showing that is the fact that the cilium has different way of disassembling. So yeah, it's formed, but it could also be disassembled. So there are different ways, and we still do not understand how these different phenomena occurs and when and what tissue. So we're trying to study all these aspects, which are very, very cool in our eyes. And so you can see clearly that the cilium here, green, will disassemble gradually over time. And you can see it's going to disappear slowly over time. So these are just videos of that. And then it's going to disassemble completely. So you can see now that it's going to disassemble so fast, and then almost like in a second, you can see clearly it's going to disappear right away. Yeah, so you have these dynamics that are still poorly understood. And so I'm going to add another layer of complexity. Yes, it has the primary cilium, which is a sensory organelle present at the surface of the cell, has these different dynamics happening, but it has also a complex trafficking happening inside the primary cilium. This is what we call molecular trafficking. And this is an illustration of the primary cilium. So this is a membrane-bound organelle in which you have microtubules, which is one of the cytoskeleton of the cell. And so the cilium will emanate from a basal body, which is a mother centriole that matured through molecular changes that I'm going to detail later on. And so you have this primary cilium that emanates from the cell. And to do so and to be formed, you need what we call protein cargoes or IFT, cargoes, intraflagellar cargoes. And these IFT, they go up and down from the lower side. They go up to the upper side and then they go down. And this is just a video to show you these cargoes going up and down in the primary cilium itself. And so this pathway is important for the cilia formation, the maintenance of the structure of the cilium, as well as the signaling output of the primary cilium. So my lab focus is to study primary cilia biology, understand how proteins are trafficking from the lower side of the cilium, going to the tip and going out. So really looking at primary cilia trafficking, closely looking at also some instabilities happening at the tip of the cilium, trying to understand what are the mechanism and molecular pathway that regulates this and how it's disrupted and the disease aspect. I'm sorry, I'm going to use the word mutations, but this is just to emphasize we're looking at changes or genomic changes in patient overall. And so we're trying to characterize and understand the different celiac dysfunction that could happen in the presence of these mutations and how this in turn will impact basically our behavior as well as induce some brain dysfunction as well. So today I'll be detailing one of the projects that we are working on. And so in this project, we would like to investigate all these primary cilia disruption in terms of length, trafficking. So we did some of these characterization, and I'm going to show some of that, as well as maybe also detailing how we arrived to a point we are studying this kinase. So the first part would be really showing some published data about the function of the kinase CSMK2 alpha 1 and its role in primary cilia trafficking and stability, but also allude a little bit on the phenotypes that are related to cilia and the disruption we saw in this organelle in OCNDS patient cells. And so we are studying additional aspect of that. And our ultimate goal is to identify proteins that are disrupted or perturbed, or like substrate that are disrupted and perturbed in OCNDS patient cells, and to try really to link these disruptions to the primary cellular function, and why not to the underlying molecular disruption in OCNDS. And of course, this is using different cellular models that we are using, like mouse embryonic fibroblasts, as well as OCNDS patient cells. So how did we come up with this CSMK2 alpha 1 and basically study this in primary cellular biology? So I'm going to try to summarize the rationale of how we studied or we identified CSMK2 alpha 1. And so we were studying at the time this tautoglin kinase 2, which is a kinase required for cilia assembly. And so this kinase is important for the recruitment of IFT cargos, as well as the removal of an inhibitory protein called CP110. So these two events are critical for cilia formation. So at that time, we don't have a good idea about the molecular pathways through which TTBK2 is performing these different actions. So what we did is basically perform two unbiased screen that I'm not going to detail today, but these unbiased screen will allow us to identify CSMK2 alpha 1 in both of them. And so one screen is about, which is the proximity dependent identification to identify potential interactors, as well as a suppressor screen based on the mouse ganglion CRISPR knockout library. And it allowed us to identify functional interactors of TTBK2. And so we were lucky enough to find that CSMK2 alpha 1 is mainly the only protein that is present actually as a potential interactor of TTBK2, but also a negative regulator of TTBK2 at the sonic hedgehog pathway. So, and at that time, around 2018, we found out that this kinase is actually mutated in awkward chunk neurodevelopmental disease. So I wanted to follow up on this kinase and try to see where exactly it's localized in the cell. So I'm going to make this as simple as possible. I said that TTBK2 is present at the centrosome during cellulogenesis. Well, this is how it looks like at the centrosome. And so when I stained CSMK2 alpha 1 using antibodies, primary antibodies, and secondary antibodies that will detect these primary antibodies, you can image those using microscopy. And so what we saw is basically these two proteins are partially colocalized. And I'm going to expand a little bit more on how these are organized together. So, of course, what I'm showing here, oops, we can go back. So I'm showing here, what I showed before is low resolution, I would say, images. Now we go high resolution or super resolution microscopy. And you can see a nice CSMK2 alpha 1 being enriched on one of the centrioles, which is the mother centriole. If you remember well, there is a mother and daughter centriole the mother centriole is from which you have the formation of the primary cilium itself. Okay, so we found out that CSNK2 is forming this nice bar-like structure, and it's also present at the base of the cilium here in green, and this is CSNK2. So it has a very organized structure, and I'm showing it here as just an illustration. So we wanted also to stain TTBK2, which is also present as almost a dash. And we wanted to have an idea, if we look from the upper side, TTBK2 will form actually a ring. So you can think of this as like a ring as a structure. So this is TTBK2 is forming a ring, but also CSNK2 is forming almost an overlapping structure that is partially overlapping or like localizing with TTBK2. So you can imagine that CSNK2 is actually forming a ring. So you have the cilium, and then CSNK2 is having a ring and is also forming this battery-like structure at the base of the cilium. So it was an exciting finding, and this is just an illustration. We were super excited about this. This is just to, at that time, we were able basically to look into what's known in terms of function. There was nothing known in terms of cilia function. This kinase is important for cellular growth, proliferation, and apoptosis. In the mouse model, it's lethal by embryonic stage 11.5 with defects in normal cardiac limb development. And so these phenotypes are really could be related to cilia defects. And of course, it's the gene is mutated in OCNDS. So we wanted to study further the primary cilia biology of CSNK2 alpha 1. So we removed it from the cell. That's what we call a knockout. And what we saw is we did not affect cilia formation. So the cell without CSNK2, they still form cilia. However, we saw that it's basically it formed longer cilia. So you can see quantification here. This is just data points of many cells that we quantified. And you can see clearly that the length is slightly extended. So this may mean that the trafficking that we talked about in the beginning is also affected. So we tried to stain for different ciliary proteins. This includes IFD proteins, signaling proteins like the sonic hedgehog pathway. What we saw so far, as to make this brief, is that without CSNK2 alpha 1, the primary cilium is not normal. It's longer. It has more IFD cocos. It has more signaling molecules of the sonic hedgehog pathway, like smoothened GLI2, and I'm showing these only today. IFD81, which is one of the cargos, it's actually enhanced, so you have higher intensities of that cargos inside the cilium. In control, is supposed to be just at the base, at the tip of the cilium. But if you look closely in the knockouts, I'm showing two examples for each of the protein, for each of the conditions, and you can see more enrichment or higher enrichment. And I see that for many IFD proteins, as well as sonic hedgehog pathway, including the signaling molecules, smoothened and GLI2. Another phenotype that we were able to see is we imaged these cilia. In control conditions, you have wild type or normal CSNK2 alpha 1, and this is cell line without CSNK2 alpha 1. And so if you go and image those, we take an image every minute, and you can see the base and the tip. And this is a control, so you don't see any dynamics or breakages happening. But if you look at the cell without CSNK2, you can see clearly that the tip is breaking off. So it has these dynamics or like more breakages happening. And we see that 3 times more in these knockout cells. I am not detailing or presenting all the paper that we published in 2021. Please take a look at this paper. It's open source, and I will be happy to respond to any questions. So in this paper we published and we discovered that CSNK2 alpha 1 is important for cilia trafficking and stability, and we also link it to disruption happening in OCNDS. So what we found is that CSNK2 alpha 1, as I said, is forming a battery-like structure at the mother centriole or the basal body, at the base of the cilium. It's playing as a fail-safe mechanism where it's inhibiting the accumulation of unwanted actin modulators. within the primary cilium. It's negatively regulating TTPK2 as well as the sonic hedgehog pathway. It's modulating the trafficking within the primary cilium, especially the entry of proteins within the primary cilium, and finally, the stability of the tip compartment. So, and since the gene is mutated in OCNDS, and that this disease has some overlapping phenotypes, especially in the brain, that could be related to typical ciliopathies. So we wanted to investigate OCNDS patient cells. They have disruptions in primary cilia function and regulation. I'm not going to expand too much on this slide, but I think there are three takeaway messages that I want to communicate. Is the fact that the mutations affecting CSNK2 gene will lead to neurodevelopmental abnormalities with a variety of clinical features. The second aspect is the fact that we don't know exactly the underlying molecular and cellular mechanism that lead to this disease, and that's why we are trying to characterize those and investigate those as we go. With DG and CK2 Foundation, we were able to obtain 6 cell lines to control ones from unaffected individuals and 4 patient cells with 3 different genomic changes. And keep in mind that these changes basically are heterozygous, meaning that it's just one of the copy of the gene is affected, and have 3 different type of genomic changes. Before diving into the results, I'm very grateful to the patient cells and unaffected individuals who donated these cell lines, and I would like to thank them for donating these precious reagents. So we're trying to work hard to characterize these primary cilia disruptions in these patient cells, and I'm very grateful for these patients. So what we did is we plated these cells on camera strips, and we stained them with different proteins of interest that are related to cilia. And so what we did is measure cilia length, as I did with the knockouts and CSNK2 alpha 1 story that I presented before. And so you can see clearly that all patient cells are showing shorter cilia compared to the control. So the control cell line is actually showing a higher length. So we have a consistent phenotype between patient cells, even though they come from different individuals. So this was very striking. We saw the same thing with the signaling molecules, the sonic hedgehog proteins within our receptors within, and we see lower levels of this protein compared to the primary cilium, controlled primary cilium. And so this is the quantification we saw. We did for student, and we did not see any, or like maybe one of the cell line was showing some cilia formation defects, but the others did not show any defect for on the percentage of cells forming primary cilia. So we think there is a clear ciliary defects in OCNDS patient cells, and maybe there is a disruption of the ciliary function of this kinase in these patient cells, or in the presence of these OCNDS variants. Then we wanted to look at the signaling related to primary cilia. So we evaluated the sonic hedgehog induction or activation in OCNDS patient cells. And what we see is perturbed induction or activation on all patient cells compared to the control. So we were comparing for batch 1, which is a sonic hedgehog target gene, and we see higher induction for all patient cells. So we're comparing this, and this. So everything with induction to this bar, and we see clearly higher mRNA fold change for batch compared to the control. On the other hand, we see a reduction of CLE1 at the basal level for all patient cells. So we're comparing this bar to this bar here, this bar here, this bar here, and this bar here. So there is clearly sonic hedgehog perturbation in OCNDS patient cells, and usually it's related to an abnormal signaling of the primary cilium. So there is clearly ciliary phenotype in these OCNDS patient cells. So the next step was to look into the CSNK2 barrel-like structure in these OCNDS patient cells. If you remember well, I showed that the CSNK2 or this kinase is forming this barrel-like structure at the base of the cilium. So what we did is stain for the CSNK2. On the other hand, you should just know that in patient cells, we have both a normal version and a changed version. So this antibody will be able to detect both, right? So in green, you can see clearly in control the normal structure of CSNK2 alpha 1. And I'm showing 4 examples. And in patient cells, you can see clearly that either it's disrupted, not present, or clearly disorganized at the base of the cilium in one of the patient cells. So while we looked, I would say, in the other patient cells, and we see the same phenotype over and over. So normal cell line, normal structure in the control cells, but disrupted in the other patient cells, as you can see here for the amino acid 156 as well as the 198 amino acids. You can see clearly half of the barrel-like structure or like disrupted organization. That's what we are seeing. So we think that CSNK2 alpha 1 enrichment at the base of the cilium in OCNDS patient cells is abnormal and it's disrupted. And this may lead to disruptions in the ciliary functions of this kinase. Well, after characterizing these different cilia disruptions in OCNDS patient cells, what are the next steps? And let's take some steps back and then think about this logically and how these point mutations of the CSNK2 gene would affect basically the protein function itself. So we think that there is 2 effects that could happen if these mutations are in effect in these patient cells. It could have a reduction in the kinase activity. So basically, an abnormal phosphoproteome. Phosphoproteome would be the number of proteins, or like the proteins in the cell that are actually phosphorylated or modified by this kinase. So we may have a kinase activity that is reduced, but we may also have changes in the 3-dimensional structure of this CSNK2 alpha 1, which may also lead to protein-protein interaction that are changed. So we may have 2 things happening at the same time. And I think to address that, what we wanted to do is to characterize the shifts in phosphoproteomes between all OCNDS variants and the 2 control cell line we have. And so since we are focusing on the centrosome ciliary compartments, we would like to overlap the hits that are, or the number of proteins that are perturbed in these patient cells, and overlap that with the proteome, or the number of proteins we know that they are localized to ciliary centrosome compartments, since we think that the primary cilium is disrupted in these OCNDS patient cells. And so basically this is to narrow down the number of proteins, since you get a lot of hits from such a screen. And so this is a way of prioritizing the study. And so to identify these hits or proteins that are perturbed in OCNDS cells, in all OCNDS cells, compared to the control cells, what we did is a quantitative phosphoproteomics using GMT multiplex proteomics. And so we used 6 cell lines to control, and then the 4 OCNDS patient cells. We plated them and serum starved them for 72 hours to form primary cilia. And then we sequentially collected from 3 independent experiments. So we have 3 biological replicates. And so we did 8 comparisons, overlapped them, and were able to identify 2 overlapping hits that are enriched or hyperphosphorylated, or 2 overlapping depleted proteins or hits that are hypophosphorylated. And so I'm not presenting the data today about these proteins, but we decided to move forward with the hyperphosphorylated as they were more robust hits. So let me now summarize basically what we have found about the primary cilia in OCNDS. We think that there is a clear phenotype of primary cilia in OCNDS patient cells. We think that these cilia are defective, and it may be be explained by the fact that CSNK2 is no longer well organized at the base of the cilium. We think there are defective trafficking within the primary cilium in OCNDS cilia. The two hits we identify are actually centrosomal proteins, so we think they could be the underlying molecular mechanism that through which CSNK2 or mutated CSNK2 alpha 1 is disrupting the primary cilium. We'll be using mouse genetics to try to study primary cilia and neural tube patterning as well as brain development using the mouse model. And so depending on the funding we'll be getting, we'll generate additional mouse model for other OCNDS variants. At Sanford Research, we have the ability to do that, and it doesn't cost that much. It needs just more, we have the technical skills to do it, basically. And so finally, for the clinician in the room, yes, OCNDS is not a typical ciliopathy. It is not a typical ciliopathy, but there is a clear trend right now showing that many neurodevelopmental disorders are actually linked to cilia disruptions. And since the brain is still a black box for the different roles of primary cilia in the brain, so we should be careful a little bit here and try to investigate further this in vivo. And that's what we intend to do to really understand the ciliary disruption in the brain in OCNDS mice. So that's our goal, and we'll be also trying to do some rescue of the behavioral phenotype that we are seeing in the 198 mutation, for example, or if we generate other mouse models. Well, thank you so much for your attention. I'll be happy to respond to any of your questions, and thank you very much.
All right, everyone. So I know that was a lot of information about cilia today, something you probably didn't know about before. We do have one question that popped up in the chat since we have you live, Dr. Wilsey. It's from Catherine, and I agree with her comment. She says she's struck by how many single gene disorders have overlap with the cilia phenotype and expression. So if you could speak to we as patient advocacy organizations can help to collect information that maybe could lead to insights in how we overlap clinically or by treatment, because by grouping together, that we could really push forward for funding opportunities. And she did mention things like looking through clinical records with an eye to some of these ciliary defect phenotypes. And then of course your nitric oxide testing. So any insights you might have as to how we can work together on that?
Yeah, 100%. It's a great question. Clinically, cilia are still kind of not mainstream. And I think the first step is always just to establish that the gene has a bona fide role at cilia. So I think that's certainly the case now for CSNK and 2 alpha 1, right? So that's the first step, is just sort of what happens to cilia in the context of these variants. I also think, medical record data can be really helpful. A lot of times you'll talk to foundations and they'll say, no, there's no congenital heart disease. But then you look back at patient medical record data, and for sure there's, 20% have heart murmurs, and it's just not discussed because there are bigger issues at hand to discuss clinically, right? And so I think there can be hints there in the medical record data that can support the idea that cilia are involved for sure. But ultimately, I think the cellular and animal models are going to be where people really can firmly establish that cilia are involved. And then I think it becomes, it is striking how many of these genes have a ciliary function. I don't think they all do the same thing at cilia, and I was struck in that second talk how in some models the cilia get longer and in other models the cilia are shorter, right? So I think there's a lot of cell type specificity and interactions with different variants might cause different effects on cilia. And so of course it's going to be complicated, but I think if one could find multiple genes that had the same similar effect on cilia, for example, either making them shorter or a group that makes them longer, I think there could be strength in numbers, grouping together genes like that, and then doing drug screens in in cellular models, cilia, you can do automated phenotyping and length and percent ciliated pretty easy by microscopy and culture. And then you can just add, repurpose FDA approved drugs on top of that and try to reverse those cilia issues using a number of drugs. And so we're certainly thinking about that, doing that kind of thing in my lab. But again, it's which cell type to do it on, and that gets complicated, and what kind of variants to do it on. But I think there could be use there because cilia are pretty straightforward to phenotype, to being able to do large scale drug screening on cells to start to get some hits. For example, lithium is a drug that actually lengthens cilia. So for me, that's when I would prioritize in these cellular models of variants to see if the lithium can actually help lengthen those cilia or not, right? And so lithium is super safe, used drug forever, for mood stabilization. And so that might be an interesting one to take forward for testing drugs and things like that. Nasal nitric oxide, I'm on the fence about whether or not that's going to be helpful. I think it can certainly be helpful for establishing that there's a ciliary component to that gene. I think that's very strong evidence that the gene is involved in ciliary infection. I don't think that the magnitude of changes we've seen so far are such that it could be diagnostic, for example, or resolve variants of unknown significance or things like that. But I do think it could help in that first stage of, does this gene have a ciliary component, which I think ultimately is probably the biggest intellectual leap forward in this space that could happen for genes where it's really there's no clear link yet. Those are my thoughts.
Awesome.
A little rambly.
Yay. No, but it helps us. Thank you, Dr. Wilsey, for your time, and thank you all for joining us. If you have further questions, please feel free to email me. Otherwise, have a great rest of your weekend.